quiz next week is you'll identify a blood cell when you see it on a slide. In addition, do the stuff that's listed in your packet. You have to tell me how many there are, what their job is. So you're going to memorize a bunch of stuff in your packet in addition to looking at it. You want to go to JDoc and make sure you read what this guy is saying, or girl, because they actually walk you through really easily what the different blood slides are doing and all that. So I'm just going to walk you through some slides at random from JDoc. Here's the most classic view you'll see a neuroscope come next week your exam. So you're going to look at that and say, Mr. Christie, what do I see? First thing you're going to identify are these. Red blood cells or erythrocytes, whatever. Then you identify these. Platelets, those are the easy ones. Then you got the white blood cells you have to identify further. So white blood cells, you want to remember, never let monkeys eat bananas. That's the order. So Neutrophils Most common, lymphocyte, monocyte, eosinophils. So if in doubt, it's going to be a never or let. Those are over half of the cells. So if you just remember the order, you look, well, just statistics say it should be a neutrophil in there somewhere. So neutrophils look like this. How would you identify? How would you say? What do you see there?
Multiple nucleus.
Yeah, multi-lobed nucleus. It can be 2, can be 3, can be more, but they have this lots of little lobes, and they're considered a granulocyte, even though you can't see the granules per se. They should be spotted. And that's why they have an ending of fill. If they say fill, they have speckles. If they say cyte, they don't. So granulocytes are the fills. Granulocytes are the cytes. So this is a neutrophil. You can look carefully. There's speckles in the cytoplasm of the cell. They're very faint. But they're speckly. They're granulocyte, and those are neutrophils. This one is not a neutrophil. That is a what? Lymphocyte. That's the left.
Maybe that's
It's a
center
cell,
one.
it's a cyte, so it doesn't have speckles. The nucleus is basically the whole cell. You don't see much cytoplasm. So the trick on a lymphocyte is just a dark purple circle. Flash for a slide. Reds,
Perfect, thanks.
platelets, lymphocytes, neutrophils. You okay? So you're going to look at the nucleus, you're going to look at the speckling, kind of the overall shape. Problem is they vary a lot. Neutrophil. This one down here. Exactly the same. This one. The lymphocyte. Those are the most common ones. Neutrophils, multi-lobed nucleus. Maybe you see speckles. Lymphocytes, all going to be dark. Very little cytoplasm. So those are that's the 2 most common ones. So if you that's a monocyte. This is actually one of the harder ones to name because they vary a lot in their size and shape. Your book will say they're always bigger than the reds. That's normally true, but not always. So don't use size as the sole criterion. So what you want to do is look at the nucleus versus the cytoplasm. Normally the nucleus in a monocyte is about half-ish. So you can see there's a lot more cytoplasm in the lymphocyte. It usually is a horseshoe or a kidney bean, not always. But you look at that and say, that doesn't look like a lymphocyte, and it doesn't look like a neutrophil. Therefore, it's going to be a monocyte. But personally, these ones are always hard because they vary a lot more than the other ones do. They look for size, along with nucleus, along with no speckles, along with cytoplasm size. That's a monocyte.
So it fills up less of the cytoplasm than the lymphocyte.
Right. So I'm not an artist. I know that.
You see more of the cytoplasm.
So a lymphocyte normally, you'll see all the dark circle and a little sliver, like a little moon of cytoplasm. That's the lymphocyte. In the monocyte, you'll sort of see, you'll see more cytoplasm with the nucleus. The nucleus may be bigger than the lymphocyte, but you'll see that it's not all the cell that you see traditionally. Let's talk about speckles. Let's find a speckle you want. Anyone find a granulocyte for me on this picture? Right here. This is an eosinophil. It's going to be, that's granular, lots of speckles all over. Normally these are reddish, not always, so don't always say the red one. Normally they are. And if you can look at the nucleus, it has this weird sort of figure S, bilobed, kind of ampersand sort of shape. But it's going to be speckled enough that you'll notice it. You'll see the dots. Okay. So up here is also a granulocyte. Who's that one up there? That's a neutrophil. So we're going to fill. But this one you can see has a lot less speckles. This one has a lot more speckles. But they're both fills because they're both granulocytes. Right? He's the second to most rare. All right, so this is using Boston. Let's go through these are all the granulocytes. I can tell they're speckled. Let's do Mr. Easy down here. Neutrophil. This one, that's neutrophil. Even though it's not red, you can see the speckles in the nucleus. Look at that beast up there.
Basophil.
That's a basophil. They're very, very speckled. In fact, they're so speckled, you can't see anything but speckles. Right? So you won't see a nucleus. There's one in there. But it's going to be just ink blob kind of shape. Very distinctively dark speckles. That's the most rare, less than 1%. Hint, hint. In my years of doing this, it's very hard for me to find one of those on a slide if I don't want to spend 3 hours looking for it. So the chance of me finding one on a slide and putting it on your test is based upon how much time I want to be here, which is not as much as you think. So I rarely put basophils on a slide. It just takes me an hour to find one, unless I'm lucky. The chances are neutrophils, lymphocytes, monocytes, those I can find in 2 seconds. Eosinophils take me about 20 minutes. That one takes me longer. Just because it's the most rare, it's unlikely I'm going to see it on a slide. Right? But you should still know it. Just realize, don't keep saying basophil on your exam. Probably not the one I found.
So
Go for it.
that one is a neutrophil, but it looks oddly like an eosinophil, the other one.
This one?
Is it because the nucleus isn't attached that it's not? No, yeah, no, the other one.
This one?
Yeah.
It's more granular. You can see it's more speckles in this one. And the nucleus, it's hard to see because of speckles, but it makes kind of 2 lobes shifted. A neutrophil usually has about 3, kind of like a clover leaf.
Oh, so that is an eosinophil.
Yes, it is.
Oh, okay,
This
that's
is
what I would have
neutrophil.
figured.
This is eosinophil.
Okay,
This stuff.
yeah.
Right.
So the neutrophil has more than 2 lobes?
Normally more than 2. It usually is 3, but it doesn't have to be 3. But you're going to notice it looks more like a. Yeah, it just looks distinctly different. Hard to say.